So finishing up the basics here in chapter 4, yeehaw, we'll be in chapter 5 next. And so here we're going to be talking at somatic cell division and the process then of mitosis. Why have mitosis? Well, primarily, of course, it's going to be used early in life for growth, and then after that for repair of injuries or damage. And so it's the sequence of events. We're going to duplicate chromosomes, we're going to divide those up, and we're going to produce two daughter cells that are identical to the original parental cell. So, as we've already discussed, humans have 46 chromosomes within each of their cells, or in reality, two sets of 23. One set of 23 from mom, one set of 23 from dad. So each of those sets has a number 1 chromosome, a number 2, a number 3, number 4, and on down the line until you hit the sex chromosomes. Those are for an entirely different subject much later in A&P2. But when chromosomes are similar, so you're getting a number 1 chromosome from mom and a number 1 chromosome from dad, we call those chromosomes homologous or similar ones because they have the same gene sequence on them. That's very important. They also, as it turns out, because of the same gene sequence, will be very much the same length and will be a staining procedure that can be performed and then will indicate the exact same type of staining on them. So these are ways and tricks to identify two chromosomes as being homologous. Because we have two sets in all of our body cells except in our sex cells, we call our body cells diploid, right? Di for two, ploid because there are two sets. So all cells other than the sex cells are diploid. Now, as we start a discussion of mitosis, we need to first step a little bit back and discuss what is the normal life cycle of the cell, and it's termed the cell cycle. And the part when the cell is not going through mitosis is called interphase. It has three major components in it. And the first one is called G1, the second one S, and the third one G2. We'll divulge a bit more information about those here in a bit. And the last stage of cell division is the mitosis itself. And of course, that's the subject of the later slides. So here is the cell cycle. And it begins with, we'll start at G1. And in G1, we're going to see the cell cycle is vibrant. It's producing carbs and lipids and proteins and all kinds of great stuff. And the cell's getting bigger and larger and stronger. And life is great for the cell. Then eventually it reaches a point where it's produced so much of these materials that it could divide and still be two very healthy cells as a result. And so at this point, the stage called the S phase, or the synthesis phase, will be where we see the single strand of DNA that exists at this point duplicate and become the double-stranded chromosome. So this is where we'll have two strands, they're identical to each other. Prior to this, there was just a single strand. So during the G1 or gap 1 phase, we will have only a single-stranded chromosome. But after we duplicate, now we have a duplicated chromosome, and then we enter G2. G2 or gap 2 is the point at which we are preparing for mitosis. We're gearing up, we're producing those proteins that are especially needed for mitosis to occur. Then, once that has occurred successfully, all these stages have happened, everything is great, we're successful, we're ready to enter mitosis and the 4 stages subsequently of mitosis. Now, I do want to point out that not all cells go through this cell cycle. Not all cells continue through interphase and enter the S phase. Occasionally, some cells will spin off in G1 and go into what's called the resting state, the quiescent state, or G sub 0. And examples of these cells would be nerve cells, typically muscle cells, and liver cells also will enter this state unless there is some type of incredible emergency. Then they can actually come out of it and enter the cell cycle. So, as we mentioned in the S phase, the major step that has to occur in the cell cycle during the S phase, during synthesis, is the duplication or replication of the DNA strands. And you see at the top of this figure a parent DNA double helix, and then when the command comes during the S phase for duplication to occur, these two strands will separate and you will get initiation of two more new strands being formed here. They're in the light blue turquoise that you can see there. And thus we now will have two separate strands, but they're identical to that original parental strand. So mitosis is composed of four phases. We have prophase, metaphase, anaphase, and telophase. And the acronym is PMAT, right? That works well. So prophase, what we're going to see first is that we're going to start observing that the chromosomes, which are long and thin right now, will begin condensing or shrinking into much shorter, stubbier, and broader structures that we're able to actually see in a light microscope. During the other parts of the cell cycle, before mitosis starts, you can't see chromosomes in a light microscope other than just being kind of a wispy white material like we saw when we looked at the nucleus in the cell chapter. So the chromosomes are shrinking, and now in order for the mechanisms of mitosis to work correctly, we have to have the nuclear envelope begin to disintegrate. And then the mechanisms that are actually going to separate these duplicated chromosomes, called the spindle apparatus, will begin forming. And they will begin forming from the centrioles, if you recall those from the cytoskeletal video that we discussed in chapter 3. And the spindle apparatus will consist of the centrioles along with growing microtubules. Now these growing microtubules will continue lengthening and eventually passing through where the nuclear envelope used to be, and they'll attach to the duplicated chromosomes at the kinetochores, right where the where the duplicated chromosomes are joined at. And so from one side, the microtubules from one set of centrioles will combine and join the kinetochore, and then from the other end, another microtubule will join on the opposite kinetochore. Then what they'll do is they'll start to pull on that duplicated chromosome and align it at the equator or the middle of the cell. That's metaphase, middle. Then we'll transition into anaphase, and that's when the microtubules from either side of the nucleus will begin to shrink. And as they begin to shrink, they are starting to pull on those duplicated chromosomes that then will separate at their centromeres and move towards the opposite sides of the cell. At this point, we enter telophase, and in telophase, we'll start to see some indentations occur in the cell membrane on each side, as well as the chromosomes that have been split apart now arrive at their opposite ends of the cell, and we begin to see the nuclear envelope reassemble itself. So that's the basics behind mitosis. And just to emphasize that in prophase we have a series of very important steps, and I just want to emphasize the growing of the spindle apparatus. And you can see it here. The two centrioles are eventually going to be on opposite sides of the nucleus, and their microtubules will begin growing, and you can see this in the actual photomicrograph that's displayed here. So after mitosis is completed, we've had our chromosomes arrive at the opposite ends of the cell, and the nuclear envelope is reforming. Those indentations that started to occur during telophase will continue to lengthen. This is cytoplasm division called cytokinesis. Okay, it may be just the tiniest bit observable in anaphase, but you will see it in telophase. But I also want to point out that cytokinesis is not strictly part of the mitosis process. So what develops is this structure called a cleavage furrow, and this is all regulated by those microfilaments. Remember those? They ringed around right against the edge of the cell membrane, those cytoskeletal elements. And what happens is there are anchors that extend from them into the cell membrane. So during cytokinesis, those microfilaments will begin, they're like a lasso, they'll begin to shrink. And as they shrink, those anchors they have into the cell membrane will begin to tug on the cell membrane, and they'll begin to cause this pinching in or this furrow effect. And eventually it will completely bisect the cell, and from one parent cell will produce two identical, in terms of chromosome content, two identical daughter cells. And then they're both ready to begin interphase after that. So step one, we have the production of the centrioles, the spindle apparatus, the nuclear envelopes beginning to disintegrate, and the chromosomes are starting to become visible. And this is certainly true in late prophase that you see here. And you also see some accompanying photomicrographs that are really nice at showing you what you might see in the microscope. And eventually then, as the microtubules from the centrioles make attachment, they'll start to yank around the chromosomes until they align in the middle. Then we'll hit anaphase, where we separate the chromosomes apart, but no real true evidence yet of telophase until we hit and start to see a full-out cleavage furrow. And telophase is there, you start to see the rebuilding of the nuclear envelope, and as well the chromosomes will start to relax again at this point. And then once those two cells are independent of each other, they'll both enter interphase, and eventually they both will go through mitosis as well. It's just another set of figures from an older text, and I just want to kind of bring these up to you so you start to go, oh yeah, I recognize all this. This is good. Okay, so I see interphase. I don't really have a good distinct picture of chromosomes. The two stages of prophase, early and late, and don't worry about prometaphase. I don't consider it at all. The two parts of prophase, you start to see the nuclear envelope's completely gone. The spindle apparatus is completely formed now, and we get the chromosome alignment in the middle. And then anaphase, we get the separation. And then telophase, the cleavage furrow is becoming pronounced. We're starting to get the reestablishment of the nuclear envelope. And then we get cytokinesis. So what I want you to do is to kind of look at these pictures and just kind of tell me what phase you think is being presented here. So this would be telophase. The cleavage furrow is present. You can see the line in between the two cells. The chromosomes are still evident to some degree, but they're starting to relax, and we're starting to get rebuilding of the nuclear envelope. How about this picture? So very little cleavage furrow, right? But you do have a separation of the chromosomes into two distinct bunches. This would be anaphase. In this picture, we have some evidence of a nuclear envelope present. You can see it above the chromosomes, but below them, there is no nuclear envelope. So it's already began to disintegrate. You can see the two spindle apparatuses are forming, and you can certainly visualize the chromosomes. So this would be prophase. And finally, we see chromosomes aligned in the middle. The spindle apparatus is fully developed. And so what goes with middle? Metaphase.